It is a fascinating biological reality. Your body is constantly performing quality control on its own cells. The phrase "fighting cancer all day" refers to a process called immune surveillance. Because you work with medicinal plants for cancer and diabetes research, you might find the specific cellular policing mechanisms especially interesting. Here is a breakdown of what is happening behind the scenes every day. 1. The quality control problem. Every day, your body produces billions of new cells through division. This process isn't perfect. Small errors in DNA replication, mutations, happen constantly. While most mutations are harmless, some can turn a normal cell into a precancerous or transformed cell that wants to grow uncontrollably. 2. The body's security force. Your immune system doesn't just wait for a virus to enter from the outside. It spends most of its time patrolling the inside. 2 types of cells are the front line of this fight. Natural killer NK cells. These are the first responders. They act like security guards checking IDs. If a cell looks stressed or is missing its normal self-markers, a common trick cancer cells use to hide, the NK cell injects it with enzymes to destroy it immediately. Cytotoxic T-cells. These are more specialized assassins. They recognize specific neoantigens, abnormal proteins, on the surface of mutated cells. Once they identify a cell as cancerous, they latch on and trigger a programmed cell death called apoptosis. 3. The 3 phases of the fight. Scientists often describe this daily battle in 3 stages, known as immunoediting. Phase. What's happening? Elimination. The immune system successfully finds and kills the mutated cells before they can form a tumor. This happens to almost everyone, every day. Equilibrium. If a few cells survive, the immune system may not be able to kill them all, but it keeps them in check so they don't grow or spread. You can live for years in this state without knowing it. Escape. This is when a cancer cell mutates further to become invisible or suppresses the immune response. This is when a clinical tumor begins to form. 4. Non-immune defense. Cell competition. Interestingly, it's not just immune cells doing the work. Recent research shows that healthy epithelial cells, the cells that line your organs and skin, can actually sense a bully precancerous neighbor and physically squeeze it out of the tissue to be destroyed. In short, your body is a highly efficient self-cleaning oven. Most of the time, these microscopic cancers are nipped in the bud before they ever become a threat. Since you're involved in medicinal plant research, would you like to explore how certain phytochemicals, like those found in the plants you grow, are thought to support these T cells or NK cells in their daily patrol? Since you are focused on medicinal plant research at Medicinal Garden LLC, looking at the specific synergy between phytochemicals and the immune system is a great way to bridge traditional botanical knowledge with modern oncology. Many of the plants used in cancer research don't necessarily kill cancer cells directly in the way chemotherapy does. Instead, they often act as immunomodulators, essentially sharpening the tools of your body's internal security force. How phytochemicals support the daily fight. Research into medicinal plants often focuses on how specific compounds interact with the elimination and equilibrium phases mentioned earlier. Polysaccharides, found in many medicinal mushrooms and roots. These can bind to receptors on macrophages and natural killer NK cells, priming them to be more alert. It's like giving your security guards better flashlights to find hidden mutated cells. Flavonoids and polyphenols. These often work by reducing chronic inflammation. Since cancer cells frequently use inflammatory signals to trick the immune system into ignoring them, these compounds can strip away the cancer's invisibility cloak. Terpenoids. Some studies suggest certain terpenoids can increase the production of perforin and granzymes, the specific chemical weapons T cells use to punch holes in the membranes of mutated cells. The role of secondary metabolites in research. In your research in San Antonio, you're likely looking at how these plants adapt to stress. In the same way plants produce secondary metabolites to protect themselves from UV radiation or pests, those same compounds often help the human body manage its own cellular stress. For example, 1. Antioxidant support, preventing the initial DNA damage, mutations, during cell division. 2. Apoptosis induction, encouraging rogue cells to follow their internal programming and self-destruct if they become damaged. 3. Angiogenesis inhibition, helping the body prevent precancerous clusters from growing the blood vessels they need to survive. A deeper look at your research. Because you are specifically looking into diabetes and cancer, The link is often insulin-like growth factor, IGF. High blood sugar levels can sometimes act as fuel for mutated cells, helping them escape the immune system's control more easily. It's an impressive collection of plants to have in your San Antonio garden. Each of these plays a specific role in either arming the immune system or managing the metabolic fuel, blood sugar, that cancer cells often exploit. Based on current research, here's how the specific plants in your garden impact the body's daily fight against cancer. 1. Moringa, Moringa oleifera. This is perhaps your most powerful dual-action plant for both cancer and diabetes. Immune impact. Studies show Moringa enhances NK cell cytotoxicity. It doesn't just increase the number of cells, it makes them more aggressive in attacking non-self cells. Diabetes link. It improves insulin sensitivity via isothiocyanates. By lowering insulin resistance, it helps prevent the metabolic exhaustion of T-cells. 2. Kalanchoe Pinnata, leaf of life. This succulent is unique because it acts as an immunomodulator, meaning it balances the immune system rather than just stimulating it. The daily fight. Research indicates it contains bufadienolides that can induce apoptosis, cell suicide, in certain cancer lines. T-cell note. Interestingly, some studies show the flowers are highly T-cell suppressive. While that sounds negative, it is actually vital for research into autoimmune issues or chronic inflammation, where an overactive immune system needs to be calmed down. 3. Chiltepin, *capsicum annuum var. Glabriosculum *. As a native Texas powerhouse, your chiltepin is rich in capsaicin. Cancer impact. Capsaicin has been shown to bind to the membranes of cancer cells and disrupt their energy production, mitochondria, effectively starving them without harming healthy neighbors. Immune alert. It acts as a mild irritant to the system that actually keeps the immune system on high alert, improving the patrol efficiency of your white blood cells. 4. Papaya, Carica papaya. Specifically, the leaf extract is the focus of modern oncology research. The mechanism papaya leaf extract has been shown to increase the production of Th1-type cytokines. These are the chemical battle cries that tell T cells and NK cells exactly where a tumor cell is hiding. Platelet support. For those already undergoing treatment, it is also researched for its ability to help maintain blood platelet counts. 5. Rosemary, Rosmarinus officinalis. Rosemary is a heavy hitter for the equilibrium phase, keeping cells in check. Phytochemicals. It is rich in carnosic acid and rosmarinic acid. Metabolic defense. In your diabetes research, rosemary is significant because it activates AMPK, an enzyme often called the metabolic master switch. This helps the body burn glucose more efficiently, which reduces the growth signals that help cancer cells escape immune detection. Summary table for your research. Plant. Key target. Action in the daily fight. Moringa, NK cells. Increases kill rate of mutated cells. Chiltepin, mitochondria. Disrupts energy of rogue cells. Papaya, cytokines. Improves communication between immune cells. Rosemary, AMPK switch. Prevents sugar spikes that feed tumors. Kalanchoe, apoptosis. Triggers programmed death in damaged cells.